Protein folding and simulations. The aims and objectives for this lecture are to be able to describe and discuss the importance of understanding protein folding, the problems of protein folding and the mechanisms by which they fold, the key methods used to look at folding, and some of the ways in which protein folding can be simulated in a computer. Protein folding is extremely important in disease. Things like sickle cell anemia, cancer, prion diseases such as variant Creutzfeldt-Jakob disease, Parkinson's, Huntington's, Alzheimer's disease, all are related to misfolding problems in proteins. Many of these are characterized by amyloid plaques. So on the left we have a cow suffering from mad cow disease, and this transferred across into humans as variant Creutzfeldt-Jakob disease. So these amyloid plaques are insoluble fibrils composed of misfolded proteins. They aggregate, they form deposits, and in this case affect the brain. Now, we can think of what happens to a protein in this sort of cycle here. So we start with synthesizing the protein. That then is going through a pathway to be folded up, so via some sort of intermediate state, through to the native state at the bottom here. Now, from that, we can get all sorts of things happening. So the original unfolded protein can form some sort of disordered aggregate. It can get degraded into fragments, and they can aggregate. They can form these amyloid fibrils. They can also do that via prefibrillar species. The intermediates can also go this way to form amyloid fibrils. We can get the intermediates forming disordered aggregates. The native form can oligomerize to form a crystal, which of course is what we need to happen in order to do crystallography, but we can also get association of the native forms into some sort of fibre. So a good example of that in disease is sickle cell anemia. So sickle cell anemia is caused by a single mutation in hemoglobin. Glutamate at position 6, which is shown in red in the flashing image, mutates to a valine. Now that leads to a small hydrophobic patch, and because we've got more than one of them on each individual hemoglobin molecule, because the molecules are tetramer, these can then associate with each other. So on the right the mutation is shown in green, and these can then associate, leading to these fibers or fibrils of hemoglobin, and that's what causes aggregation within the red blood cells, leading to this sickle shape of the cells and them blocking the capillaries. To understand how proteins work and cause disease, as I said in the last lecture, we really need to understand their dynamics, understand their folding, and understand their energetics. And we wish to be able to simulate all this sort of thing in a computer. So the protein folding problem really is one of going from an extended, unfolded protein to something like this, a much more compact, folded protein containing secondary structure and so on. And again, as I mentioned in the energetics lecture, this is really driven by the hydrophobic effect. So if we take that unfolded chain on the left, and the hydrophobics are shown with the filled circles, then that will try to fold up to bury those hydrophobics and leave the hydrophilic parts on the surface. Now, of course, that mechanism depends on the solvent we're talking about, whether it's water, which of course largely the inside of cells and the outside of cells is, but also we need to consider lipid bilayers, because lipid bilayers are fatty and hydrophobic, and therefore the folding, it tends to be the other way round. In other words, in the part of a protein that is spanning a lipid bilayer, the hydrophobics will be on the outside and the hydrophilics on the inside. But because of this dependence on hydrophobics and hydrophilics, other factors come into play, such as the concentration of salts, the pH and the temperature. I showed you this slide in the previous lecture as well. This is a slice through a protein, so you can imagine it like an apple that you have cut in half, and you can see from this that the hydrophobics shown in blue largely dominate the core of the protein, whereas the hydrophilics in red around the outside. It's not true to say that all proteins follow this sort of rule, though. There are also intrinsically disordered proteins, and there are many examples where functionally important proteins are partially or completely disordered. So we can have a completely disordered protein, which is an IDP, intrinsically disordered protein, and we can also have parts of proteins that are disordered, and those are known as intrinsically disordered regions, or IDRs. It's estimated that more than 50% of proteins have IDRs of at least 30 amino acids, and up to 20% of proteins are completely disordered. In some cases, ligand binding, whether that be binding to another protein or to DNA or to a small molecule, can induce a much more rigid conformation. Disordered regions tend to be enriched in polar and charged side chains. Now, of course, given what we have just said about the protein folding being dominated by the hydrophobics burying themselves away from the solvent, that makes perfect sense, because we don't have that same ability to drive the folding if we have heavily polar and charged regions. It turns out that disordered proteins very often have regulatory properties and can bind to multiple ligands. Now, if you think from an energetic perspective, we talked about this a little bit in the last lecture, when we have interactions between proteins that tend to be driven by the hydrophobic effect, just as the folding is. But if we have much more limited hydrophobics, as we tend to with intrinsically disordered proteins, then the binding energy is not going to be as strong. In addition, if we're going from a very disordered state to an ordered state on binding, then we're going to lose entropy as a result of that. So these interactions tend to be easily reversible. So when you're talking about proteins involved in regulation, where perhaps you want a temporary binding to another protein or to DNA, then having a relatively low interaction energy is something that you're looking for, because you want this to be reversible. Let's look at the problems of protein folding. These were originally recognised by Linus Pauling in the 1940s. He realised that there is a huge amount of conformational space potentially accessible to a protein. Yet there is, with the exception of the intrinsically disordered proteins, just one unique solution. And presumably this folding of proteins is rapid and spontaneous. Now, this was formalised a little bit more in 1969 by Cyrus Levinthal, who suggested the Levinthal Paradox. And this suggests that given that there's this problem, then we can actually think about the astronomical number of possible conformations available. So if we take a 100 amino acid protein, and for each amino acid just allow 3 conformations, well, that would mean there are 3 to the power of 100, i.e. 5.2 times 10 to the power of 47 different conformations possible. Now, of course, that is a bit of an oversimplification because it ignores the clashes that are going to occur between distant residues as you fold a protein into every possible conformation. But equally, it ignores the additional complication of side chain conformations. Now let's take another sort of extreme based just again on the backbone. 100 amino acids are going to have 198 rotatable backbone torsions. Those are the phi and psi angles. So if we assume we can make 5-degree steps around those, then actually every torsion angle would have 360 divided by 5, which is 72 conformations. So that means on that basis we would have 72 to the power of 198 possible conformations, which is 5.6 times 10 to the 367. Now the reality in terms of the number of potential accessible conformations is probably going to be somewhere between those two sorts of limits. Now, sampling all of those possible conformations would take an absolutely enormous amount of time, even if we sampled each conformation just once and did so every picosecond. Now, proteins actually fold much faster than this, so a random conformational search really cannot occur. We're going to take a short break at that point, and I just want you to do a couple of calculations. So first of all, calculate how long it would take to fold a protein if we visited each of 5.2 times 10 to the 47 conformations, just once each, and we visited one conformation every picosecond. That's 10 to the minus 12 seconds. Do the same thing for the other extreme that I suggested there of 5.6 times 10 to the 367 conformations. See how long that would take. Work them both out in seconds, days and years. Now, I already mentioned one thing about why reality is likely to be somewhere between those two extremes. Think about what else would limit it. If you haven't come across it before, find out about the Ramachandran plot and see how that might influence your thinking. Okay, so you should have done these calculations. So if we're looking at 5.2 times 10 to the 47 conformations, visiting one of them every picosecond, then the time taken would be 5.2 times 10 to the 47 multiplied by 10 to the minus 12 seconds, which equals 5.2 times 10 to the 35 seconds, which we can calculate to be about 6 times 10 to the 30 days, or 1.65 times 10 to the 28 years. If we do the same thing with 5.6 times 10 to the 367 conformations, then that works out at about 5.6 times 10 to the 355 seconds, 6.48 times 10 to the 350 days, or 1.78 times 10 to the 348 years. Now, the age of the universe is only about 13.7 times 10 to the 9 years, so clearly these timescales are completely impossible. Now, I also asked you to think about the Ramachandran plot and the reality being between these two extremes, because obviously just sampling 3 conformations for each amino acid is a bit of an oversimplification, but sampling every 5 degrees is probably too much. Now, the first thing, of course, that limits this is clashes between distant parts of the protein as you try to sample every conformation. But the other thing, as I said, is the Ramachandran plot, and that's different for glycine and proline and then the other amino acids. So what we're doing here is plotting the psi angle, that's the backbone angle between the C alpha and the backbone carbon, the torsion angle, against the phi angle, which is the backbone torsion angle between the nitrogen and the C alpha carbon. And we find when we do that, looking at high resolution proteins, that there are favoured areas which are shown in green and disfavoured areas that are shown in red. So the brighter the green, or the paler the green, the more favoured these regions are. So this plot on the right is what we normally see as a Ramachandran plot, and it turns out that this region around here corresponds to alpha helices, and this region around here corresponds to beta strands. Now, that doesn't mean that every time you see an amino acid in this conformation you have a beta strand, and every time you have it in this conformation you have an alpha helix. We have to repeat this conformation over and over again to give us an alpha helix, and this conformation over and over again to give us a beta strand. Now, you can see there are some other favoured regions here in green, but when we look at proline, we see that there is just a very much narrower region of favoured conformations, and that results from the cyclic nature of the proline side chain. Strictly, it's an imino acid, not an amino acid. The side chain links back onto the backbone nitrogen, and that restricts the phi angle, the one between the nitrogen and the C alpha. So you can see we don't have any of these conformations over here. In the case of glycine, of course, we don't have a side chain at all. We just have a hydrogen, and that means that we have a pretty much symmetric plot about a sort of rotational symmetry about this center point, because we no longer have the left and right-handed forms. We don't have a D and an L amino acid. So glycine is easily able to access conformations up here, round here, round here, that the other amino acids can't access. So the point of this in this context, of course, is all these red areas that individual amino acids can't adopt combinations of phi and psi angles that are in these red areas, because they lead to steric clashes with the neighbouring amino acids, or with between the side chain and the backbone. So, even accounting for all this, of course, real folding cannot take this long. So, the folding process in vitro generally takes in the order of milliseconds. When we get very small proteins, less than 100 amino acids, they really seem to fold pretty much in a single step without intermediates, and that takes of the order of microseconds. The longest time is for proline isomerization. So remember that we can get cis and trans forms of proline. This refers to the peptide bond that occurs immediately before a proline residue. Normally peptide bonds are trans, but relatively commonly for proline they can be cis. And that again is the result of the nature of the side chain. The fact that it links back onto that backbone nitrogen means that the energy difference between the cis and trans forms is much lower for proline than it is for other amino acids. Now, this is an experiment that you've probably come across before by Christian Anfinsen in the 1960s, who suggested that a protein's knowledge of how to fold is stored only in the sequence of amino acids. And he performed this classic experiment with denaturing ribonuclease. So in the experiment he took the native ribonuclease protein, and that has 100% activity. He then reduced it to break these disulfide bonds and denatured it by adding something like urea. So the denatured protein then has essentially 0% activity. If you then renature by dialysing out the urea and oxidize, then you recover about 90% of the activity. Now, as a control experiment, he did the same sort of thing. He started with the native protein, he reduced and denatured it, and found that the denatured version had essentially 0 activity. But this time he oxidized first to reform the disulfide bonds, and then dialysed out the urea to renature. And this recovered about 2% of activity. So the point of the control is to show that the protein really was being denatured. It wasn't a result of the presence of the reducing agent and the denaturant that stopped the activity. In the control, by oxidizing first and then renaturing, you're showing that much of the oxidation, the reformation of the disulfide bonds, is in scrambled versions of the protein, and only a small percent happen to be in the correct conformation for native activity. So this proved the point that essentially within the sequence of the protein there is enough information to allow it to fold up, because you weren't putting in anything extra to help the folding process. How do proteins fold? Well, there are 2 models of how this happens. The first is the diffusion collision model, where parts of the unfolded protein come together to form some sort of folding nucleus. Then there's formation of secondary structure, and finally the secondary structures collide with one another and pack together in an optimal way. The second model, the nucleation condensation model, is one in which the secondary and tertiary structures are formed at the same time. Now, clearly it's something of a philosophical difference between these 2 models, and recent studies have shown that some proteins show characteristics of both folding models. This shows the diffusion collision type of model, in which we start with a denatured unfolded protein. On the microsecond time scale we get the formation of a folding nucleus and the formation of secondary structures, and then on a millisecond time scale we get collapse of that to something that looks more like the native form, and finally on the millisecond to second time scale we get formation of the native protein. There's also a fair amount of evidence that folding actually begins while the protein is being synthesized. So this is co-translational folding, and that means that the N-terminus can start to fold while the C-terminal portion is still being synthesized. And as it happens, this is now being exploited in some simulations of protein folding, in which the folding of the N-terminus is simulated before the C-terminus. How can we observe what actually goes on in protein folding? Well, there are a number of experimental techniques that can be applied. The first of these is NMR, where we can monitor hydrogen deuterium exchange of backbone amide protons. So this protein CYLR2 was studied by Jaremko et al. And shows a progressive dissociation. So what was being simulated here, in fact, was unfolding. So we start with a dimeric, fully folded protein. That then dissociates into native-like monomers and then into a highly dynamic, partially folded state. We can also exploit CD, circular dichroism, and this is a technique where chiral structures such as alpha helices and beta strands absorb circularly polarized light. Measuring the change in the absorption of light as a function of the concentration of some denaturant or of the temperature allows us to see how much secondary structure is present. And this can be combined with fast mixing devices such as stop flow techniques in order to measure the kinetics of protein folding. So here are some examples using CD and NMR. So the top example of lysozyme, this is monitored with NMR. We see over time that beta sheets are starting to form, alpha helices start to form more quickly. This bottom example uses CD, and this is an all-alpha protein, and what we see is that the alpha helices start to form very quickly and plateau out to 100% formation here. And here we're monitoring aromatic side chains, so you can see that as the alpha helices form, the aromatic side chains are also starting to move into the correct positions. Now, the reason, of course, that this happens, that we see the alpha helices forming more quickly than the beta strands, is that they are internally stabilized. So remember that the hydrogen bonds that are present in an alpha helix are within the helix, whereas the hydrogen bonds that stabilize a beta sheet are between separate strands which have to come together in space. Now, another technique that can be used is to look at protein unfolding experimentally. Now, the experiment that I mentioned a couple of slides ago was actually about unfolding. This is another way of looking at unfolding and actually doing it physically. So in this case, the protein looked at was RNase H. What happens is that we mutate a residue near the end of each chain to a cysteine, and that is then covalently linked to about 500 base pairs of double-stranded DNA, and that's simply used as a linker. The other end of the DNA linker is attached to a styrene bead. So at this end, at the bottom, we have a bead, and that's then fixed in place by using suction onto a pipette tip. It turns out that if these beads are small enough, they can actually be moved using light. So at the other end, we focus a beam of light onto the bead, and that can then be used to move and pull on this linker. So when we do this, when we attach the DNA linkers to the RNAs H, we find that the CD spectrum is identical to the native protein, indicating that it is correctly folded and that these linkers added on have no effect on the structure. But once we've done this, we can now start to pull on this bead and stretch the protein, try to pull it apart. And this technique is known as optical tweezers. So these are the results that we get from this sort of thing. And if you look at the graph here, this is the amount of extension we're getting, and this is the force being applied. So with the protein in its native state here, we now start to apply some force to pull the protein apart, and we go through an energy maximum here. We're having to apply lots of force, and all of a sudden, with about 19 piconewtons of force here, we have an abrupt change where the protein starts to be pulled apart. And we can think of that, if we think of this sort of energy surface here, this is the native down here, and we're pulling up over this energy barrier here, and then we're dropping down again to an intermediate state, which is somewhat lower energy. We're not having to pull so hard to keep it in that form. Then we do more pulling, and you can see that the protein is extending with relatively little extra force. There's a bit of another barrier here, but we can think of this here as coming up this energy well up the side here towards this unfolded state. And this sort of finding from using optical tweezers is consistent with previous studies suggesting that there is a stable core of the protein that folds first, and that's what is gradually being pulled apart here, and there's this sort of collapse to this native state here. So let's look a bit more at this idea of conformational space and energy landscapes. So you just saw that sort of thing in the kind of funnel-shaped plot that I showed you on the last slide. So let's think about what happens as a protein folds, going from some unfolded state here to a fully folded state here. Now, we can assume that it goes through some set of intermediates. There may be only one intermediate, there may be many. Now these may or may not be relatively stable by themselves, but it may just be a kind of pathway that we go through where none of these states is stable, but we're just accessing each of them in turn. And we can also imagine that from any one of these states we can go off this proper folding pathway to some so-called trapped state, some kind of off-pathway intermediate, and we have to go back to this proper intermediate in order to get back on our folding pathway. Now, there is this concept of a molten globule. These have common structural characteristics. They have a considerable amount of secondary structure. However, most of the precise tertiary structure is missing. They tend to be relatively compact, and they have a loosely packed hydrophobic core. So the question then is, are these molten globules intermediates on this pathway, or are they representative of these trapped states? So this is an example of cytochrome B562. This is the properly folded protein on the right, and a molten globule form on the left. You can see that the secondary structures are mostly in place. This alpha helix towards the right isn't properly folded yet, but overall most of the secondary structure is there, and the arrangement of secondary structures is roughly correct. So this idea of conformational space is one in which we think of an energy landscape. Now, this is obviously a very, very much simplified view, and here we've basically only got two variables in shape going along x and y, and then we have an energy variable being plotted in z. So the simplest type of folding would represent a smooth surface like this, where we start at a high energy conformation and we just slide down this funnel to the native conformation here. In reality, of course, we have far more than the two conformational degrees of freedom. We've already talked about that with the number of rotatable bonds and so on. So in reality, this would be an n-dimensional space that we're looking at. The problem, of course, is that in reality it's not a nice smooth surface like this. It is actually a very, very rough surface like this. So what I've done here is show you a picture with a slice then taken through that surface that looks like this. And that slice shows you a kind of one-dimensional now variable with energy going this way. And what you see here is what we refer to as a local minimum. Now, that's a lower energy state. However, it's not the lowest energy state, the native structure, because that's here. So again, you can think of these trapped states as being these local minima in the energy surface. This is another representation of the same sort of thing. What we have on the left here is energy. So we're trying to get to some sort of energy minimum, which is the native folded state. Now up the top here we have lots and lots of possible different unfolded forms, which means that we have lots of entropy, lots of randomness. Now as we work down this folding pathway, we're going to go through these intermediates, which may be these molten globules, and gradually move ourselves towards this native state. And as we move through this, we go through these little trapped states that we have to come out of in order to get ourselves to the native state. This is basically the same diagram again, but here it's suggesting that these states in the middle are all molten globules. So we're going through this partially folded state in order to get to the native structure here. In other words, molten globules are clearly, on this basis, intermediates in that folding pathway. This image then reflects one view, which is that the molten globule is a necessary folding intermediate. The other view is that the molten globule is just these little regions here where we're moving off the proper folding pathway and moving to a trapped state. Now this alternative view actually encompasses both possibilities. So if our folding entropy reduces to being quite low during an early stage, in other words, we're getting some sort of partial folding, then we go through a stage in which we have a very, very rough surface of lots of trapped intermediates, and essentially we can't avoid that. This is just an expanded version of the same thing. We have to go through this molten globule state because the chance of falling right down the middle of this is very, very small. So in this case we are suggesting that the molten globule is indeed a trapped state, but we are not going to get to the properly folded form without going via that trapped state. At this point we're going to take another short break, and I want you to download the following three papers: Unachik et al. From 1997, Redfield, which is just a paper on the internet, and Bishkova 2018. Now you don't necessarily have to read these all thoroughly, but definitely do read the Redfield paper, because that's quite short. The other two, just read them briefly to get a sort of overview of ideas about protein folding energy landscapes and the history of the molten globule. How, then, can we simulate protein folding? So, as I said in the last lecture, we can put together a force field that describes the energetics of a protein in terms of bonds, bond angles, torsion angles, electrostatics, van der Waals interactions, and possibly also hydrogen bonds. So, given that force field, can we use it to simulate protein folding in a computer? Well, we can write software to simulate the movements of atoms under the influence of this force field, and that's what we do, as I described in the last lecture, with molecular dynamics and energy minimization. So this is this example that I showed you before of molecular dynamics, of atoms moving under that sort of set of forces. But it doesn't work for protein folding. So why doesn't it work? Well, it could be the accuracy of the force field. After all, the parameters come from small molecules. But really, the sorts of simulations that we can do with molecular dynamics suggest that that probably isn't a major factor. Is it our ability to explore conformational space? Well, yes, that's almost certainly part of it. As we've discussed before, there's a huge amount of space that could be explored, and we could get trapped in local minima. Is there anything else? Well, yes, it's our old friend, the hydrophobic effect. And this really is the key factor in protein folding. Hydrophilic residues, as we've seen before, shown in red, like to be on the outside interacting with water, while the hydrophobic residues like to be buried away from water. And because this is an entropic effect, it can't be simulated easily just using the standard force fields. What we can do quite well with molecular dynamics is unfold a protein. So what we do is anchor one end and then apply a force to the other end, just as we did in the real experiment with optical tweezers. So what you see here is, if we start at the beginning, the compact structure, you can see it gradually starting to unfold, and at this point you could see that there was a small nucleation area, just a little bit of structure just here, that still maintained, and after that it's just individual strands of secondary structure which eventually would get pulled apart. Now, if you imagine running that simulation backwards, this nucleation point is exactly what we saw in one of the two models for the folding of proteins, this nucleation and then collapse. Given these problems with using normal potentials, how can we improve on that? Well, the answer, as again I mentioned in the previous lecture, is to use what's known as an empirical potential. So this is derived from observation of protein structures. So we exploit information about distances between amino acids in known proteins. So in this example here, we have an alanine 4 residues apart from a serine, and we could measure the distance between, for example, the C alphas of those, or maybe the C betas, and so on. So in this case, we can compare how often we see this arrangement, an alanine and a serine, 4 angstroms apart, separated by 10 angstroms, in folded proteins compared with random unfolded chains. Now, if we apply the inverse Boltzmann equation, which relates the statistical observations to energy, and is normally used, of course, for looking at things like the velocities of atoms moving at a certain temperature, we can use a simple logarithm calculation of observations divided by expectations, and the energy is proportional to this minus log observed over expected. So if we do this for a number of different distances, we will see a curve of this sort. We'll see certain distances are particularly unfavoured, others are what you'd expect by random chance, and in some cases you get energy minima. And of course in this case, this observation of seeing an energy minimum at 6.5 angstroms is the result of alpha helices. So you can put all this sort of thing together. You can calculate for all interactions, which I'm showing here in these lines between the residues. We can also include solvation terms that look at hydrophobics being buried and hydrophilics being on the surface, and sometimes we can ignore loops like these ones in yellow because they tend to be a bit more flexible, or if they're not dynamically flexible, then their conformations in different homologs will be different, and that can confuse our statistics. If we use this sort of thing, then we have a representation of the energy of a particular protein fold that takes into account the things that we can't measure or simulate using a normal potential. So this does take into account then things like hydrophobicity. It will encourage hydrophobics being close to each other, positive and negative charges being close to each other, and discourage things like hydrophobics on the surface and 2 positive charges close together and 2 negative charges close together. But how can we actually apply this to folding? Well, we can use simplified models of protein folding. So the simplest type of model is a lattice model, in which each amino acid is simulated as a single bead or point of a finite set of types. And often that's just done with 2 types. The HP model is hydrophobics and polar. Each residue then is restricted to be placed on the vertices of a lattice, and usually that's a simple cubic lattice. Now, sequentially adjacent residues must be placed on adjacent vertices, and then a very simple energy function in this case can be used to describe the interactions between neighbouring beads. This is the sort of thing we get from that type of model. Obviously it's not like a real protein, but it does allow us to learn about the way in which proteins can be put together to fulfill those requirements of hydrophobics on the inside interacting with each other and hydrophilics on the outside. Other simplified models have only limited success, but they are starting to improve. Now, these sorts of models tend to take a lollipop approach. So you have a backbone and you have a side chain represented as a single point coming off that backbone. There have been various attempts at doing this, including David Jones at UCL, Mike Sternberg at Imperial, and David Baker at Washington University. So the animation here is from David Jones. We're starting here. You can gradually see we're starting to nucleate, form little compact regions, and at the end here we're just shuffling around a little bit to find a good conformation, and we're just spinning it around. Now, these methods are quite effective now for folding small proteins, less than 100 amino acids or so, but above that it starts to get more difficult and the results are not as good. Now, Mike Sternberg in his group at Imperial has started to use things like simulating from the N-terminus that I mentioned earlier. So this is effectively like looking at the folding as a protein comes off the ribosome. His group has also started to look at the fact that proteins, when they're synthesized, are not synthesized into a vacuum or even just into water, but into an environment where there are lots of other proteins, salts and so on. And so if you can simulate the sort of pressure effect that those have, he seems to be getting better results in protein folding from doing that. Now, there are also some folding experiments that you can take part in. So folding at home is a simulation of protein folding, and they've shown that most of the time involved in protein folding, up to 96%, is spent in intermediate conformational states. And these are shown here. We're starting with an unfolded protein, trying to get to this properly folded protein at the end, and we're going through all these intermediate states, which presumably are some sort of local minima. Now, folding at home actually uses these as starting points for simulations. It describes conformational and energy landscape as a set of distinct structures and short transitions between them. Now, this idea of short transitions and being able to start from lots of different kind of starting points here means that you can split this folding task up very successfully and run it across lots and lots of different computers. Now, folding at home, which I show at the bottom here, you can go to their website, you can download their software, and you can just run it in the background on your computer. It has lots of options, including whether it just runs when you're not busy, so a bit like a screensaver, or whether it runs all the time, but just at low priority, or you can just switch it off and switch it on, and you can say how much of your computing resources you want it to be able to occupy. So I've been running that for several months now. You don't have to do anything to fold a protein. They just automatically send jobs to the system, and you just pick them up without having to do anything at all, except for leaving this running in the background. I've been running this, as I said, for several months, and most of the jobs that come to it are involved in folding proteins related to COVID-19. So obviously this is a very important effort to which we can contribute. The example at the top is another one, Rosetta at home. This is a very similar sort of idea, and again you can download software that sits in the background and does protein folding for you. Let's move back now from simulations to protein folding in the cell. You've seen this slide before, where in vitro we talk about small proteins folding pretty much in a single step on the microsecond timescale, most proteins folding on a millisecond timescale, but some proteins, particularly those that involve something like proline isomerization, take hours to fold in vitro. In the cell, these things will still happen much faster. Now, why is that? Well, it's generally as a result of the presence of chaperones. So chaperones are proteins that are associated with a protein during part of the folding process. Sometimes several different chaperones are required. For example, in the folding of rhodonese, we start off with our protein. It interacts with a chaperone called DNA-K, and then another protein chaperone called DNA-J comes along. Those come off, and GrowEL and GrowES interact instead, and then the GrowEL and GrowES are lost to leave the folded protein. The main function of chaperones is to prevent aggregation. So remember, when the protein is unfolded, there are significant hydrophobic regions that are going to pack inside. And if those are exposed, as we saw on that kind of flow chart at the beginning of how proteins can aggregate, then these unfolded proteins are likely to aggregate. So by trapping them in a hole, and these chaperones often have donut-like holes, you protect the molecule from others with which it might aggregate. Now this has an interesting side effect, which is that the chaperones in mammalian cells are often different from the chaperones in bacteria, and that can mean that as a molecular biologist, producing mammalian proteins, expressing mammalian proteins in bacteria can be difficult because they aggregate because the chaperones are different. So this is GrowEL and GrowES. In the middle slide here you can see two subunits of GrowEL and one of GrowES here in yellow at the top. And this sort of slice through that image shows you that there's a hole here with a cap at the end, and a protein can enter here, and through a series of conformational changes we get a cap going on and a cap coming off, and the protein is able to fold in that local environment without interactions with other proteins. In summary then, we've looked at why understanding folding is important, its relationship to various diseases. We've looked at some of the problems of protein folding from a theoretical point of view. We've looked at how proteins fold, some experimental techniques to observe folding. We've considered the problems of conformational space and energy landscapes. Then we've looked at simplified models of proteins in order to look at simulating protein folding because of the fact that the standard potentials really don't work. Simplest models were those lattice ones, and then we have slightly more complicated ones that are much more successful in real protein folding, these lollipop type models. And finally we looked briefly at protein folding in the cell and how chaperones can speed up this process by avoiding aggregation. And finally, a couple of bits of further reading. Lesk, 3rd edition, pages 356 to 386. Then a couple of websites which do have some good information. Currently, at the time of recording this, the second one wasn't available. There seemed to be a problem on the website. I don't know if that's coming back or not.